Hello, everyone, and welcome to Charting the Future of Mental Health Precision Medicine with Proteomics. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Olink. To learn more about our sponsor, please visit olink.com. We do encourage you to participate today by submitting any questions you have during the presentations. To do so, simply type those into Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You can also submit any technical issues you have, and we'll take care of those via the text response feature. I'd now like to welcome our speaker, Clarissa Yates, CEO and founder of Ketim Therapeutics. For a complete biography on our speaker, you can click on the biography window on the left side of your screen. Clarissa, you can now begin your presentation.
Thank you so much, Matt, for the introduction. So thank you so much for joining me today. Good morning from where I am. I'm really excited that you get to be here with me today, where I share some of the very interesting data that's coming out of Ketim Therapeutics. So as Matt said, we're charting the future of mental health, precision medicine with proteomics. And this is a pilot study using Olink proteomics, novel biomarkers in postpartum depression. So a little bit about Ketim Therapeutics. So Ketim Therapeutics was founded just last year. We're based in Queensland, Australia, and we're really focused on blood biomarkers and novel therapeutics for mood disorders, with our initial focus on postpartum depression. Our approach uses next-generation protein profiling technology, combined with AI and machine learning models to predict and personalize precision medicine for PPD. Our vision is to revolutionize mental health care through precision medicine, leveraging personalized biological insights to optimize treatment and outcomes. And our mission is to collaborate, innovate, and translate current practices into novel strategies to diagnose, prevent, predict, and treat psychiatric conditions. Our strategy is based upon our 4 pillars, which is to discover any biomarkers and biological mechanisms that we see from different cohorts and studies that we do, to design the best experiments that we can to validate those biomarkers, to develop our ideas and to develop our pipeline, as well as then to deliver into clinicians and into the hands of patients. So before I begin, I'd love to just go through a little bit about my journey of my desire, my discovery, and the dream of how I went from being diagnosed with postpartum depression myself to wanting to discover the neurobiology of depression, and then really to do a startup to challenge our current beliefs and our biases. So I'll start with my desire. So my desire is really to help others who are suffering. As I mentioned previously, when I gave birth to my children, I was diagnosed with postpartum depression. It was a very difficult time in my life where I found it really difficult to find the best treatments and, being able to go through that process of not knowing whether I should be taking antidepressants or not. But eventually I did, and I found that, I was able to recover from postpartum depression. But it really got me thinking about the people who are suffering and the people who don't respond to antidepressants, which I'll go through a little bit about later. But during my time, I did actually start a company called Hooked in a Box, which was really a entrepreneurship e-commerce company looking at gift boxes for moms and babies. So I've been wanting to help moms and babies for quite a long time now. And this has led me to really go into that, desire to understand the neurobiology and start Ketim Therapeutics. So I'll go a little bit about major depressive disorder. So major depressive disorder is a very debilitating chronic mental disorder where about 1 in 5 adults in the United States will experience mental illness in any given year. The World Health Organization has predicted about 300 million people suffer from the disorder, and that is going to be the largest contributor to health and disability by 2030. So the common symptoms of depression is basically having 2 weeks of these symptoms that we see here. And the goal is really to normalize mental illness, being able to speak more openly about the condition, discussions about the work in your workplace, talking about therapy, and saying no and not feeling guilty. So this is really my inspiration to wanting to understand what it is about the biological mechanisms of major depressive disorder and why antidepressants may work for some and not for others. And the reason for that is that obviously, major depressive disorder is a very heterogeneous mental disorder, which has a lot of biological factors. And obviously, as we know as well, it's not just the biology, but we also have the environmental factors and the social factors. But in my PhD or in the work that I did later on, I really wanted to understand the biological mechanisms and how all of this have a play in the pathogenesis of major depressive disorder. And as I mentioned previously as well, we do have a very high rate of people who don't respond to their first line treatment of antidepressants. And this is something that is a real concern. So there are people out there who, would try antidepressants and it may not work the first time. And it's a huge trial and error approach to be able to find the right medication. So really it's about not being able to also identify who's going to benefit best from medication. We know as well that there's a problem of obviously not having any diagnostic or biomarker test to diagnose, and that this treatment for major depressive disorder and other mental disorders is a one-fits-all approach, which is something that doesn't quite work. So I previously mentioned as well, there was a big study done, the sequence treatment alternatives to relieve depression, the STAR*D trial. And in this trial, they were able to identify and show that in a huge real-life cohort of participants that had depression, went through a regimented step-by-step sequential treatment with antidepressants, and then stage 1, stage 2, stage 3, where they basically were given different classes and augmentation to study the response. And you can see from the graph here that, when they commence treatment, you see a benefit. Most patients will see a benefit roughly around week 4. But and you have in the green curve here, you see a complete response, and that's usually identified by their, depression scores. But you might see as well quite a large population of them will also have a partial response. And you can see in this red line here that even by week 12, and even after multiple trials of different antidepressants, there still isn't any response. And so this is really difficult in treating depression. And that's one of the reasons why, we haven't really gotten very far in terms of novel therapeutics, as well as identifying how we can treat the disorder. And really, I do believe that biomarkers is the key to diagnosis and treatment response. As you can see, it's not just about what's happening in the brain, but we have all these other biological factors that are happening within the body, such as stress and metabolism, inflammation, other than your genetics, as well as all these other factors that may actually influence the pathogenesis of major depressive disorder. So one of the things I really wanted to do after my own recovery from postpartum depression was to go on this path to discover how we could understand depression better, and how I could possibly understand what was happening when it comes to antidepressants and why antidepressants work for some and not for others. So as I said, I really wanted to understand the biological mechanisms of mental illness so that I could help others. So a little bit about the neurobiology of depression and stress. So it's very well known that stress is a huge contributor for mental disorders. And we know a little bit about the hypothalamic pituitary axis, adrenal axis, which is your HPA axis. And this is a stress response that we see in the normal people in healthy controls, where when you face a stress stimuli, your brain goes through this cycle of being able to release cortisol through CRH and ACTH, releasing stress hormones or cortisol into the bloodstream. So in a normal situation, when the stress stimuli is removed, this feedback will basically go back to normal, and you just go about on your daily life. But for some people, they have a susceptibility to this stress response, and even when the stress stimuli is removed, the body still thinks it's in stress. So you get this hyper activation of these inflammatory, not just inflammatory cells, but all, holistically in the body. And the body doesn't seem to be able to cope. And we know in the literature and research that seems to exacerbate or lead to at least the pathogenesis of depression. So during my work, my previous work, I really wanted to identify all the different models we could use to understand the neurobiology of depression. I looked at cell models, where I looked at cultured neurons, I looked at animal models, and I looked as well at real human samples. And this gave me the ability to look into the insights on the biological mechanisms of what was happening in depression. And one of the things that we can do, from a scientific point of view, a biological point of view, is identify the regions in the brain that is affected by depression. We know in the literature that the prefrontal cortex, the hippocampus, and the amygdala are the regions that are most severely affected when it comes to depressive phenotypes. We know that the decision-making processes in the prefrontal cortex is impaired in patients with major depressive disorder, where we see a reduced volume and activity. And we know that we also see a deficit in the hippocampus volume, which is to do with your learning and your memory. And we also see effects in the amygdala, which is responsible for your fear and your emotional memories, your sensitivity to stress. So we know from a neurobiology point that some things are happening within the brain that leads to the symptoms that you see in depression. We can mark these regions in animal models and isolate these regions and be able to identify and study what's happening at the molecular level in these brain regions. And this is something that we can then associate with stress phenotypes that we see in animals. And that's one of the ways that you can look at identifying the neurobiological mechanisms of mental disorders. So based upon my own background, I really wanted to understand postpartum depression, because I know that it's something that is very underlooked and very undertreated and underdiagnosed. We can see here that postpartum depression is a mental disorder that is actually defined as a major depressive disorder within the peripartum onset. So postpartum depression affects 1 in 5 women. It's quite prevalent. And in a study that looked at 565 different studies across 80 countries, over 1 million women, they found that there was a world prevalence of about 17.22%. In some of these places that they identified, especially in the lower socioeconomic countries, the prevalence of postpartum depression or PPD can go up to 60%. And so that's a really terrifying number when you think about it, because we know that with postpartum depression, if it's untreated and undiagnosed, there's a huge effect not just on the mother, but also in the family and the community as a whole. We do see a significant amount of maternal deaths in the perinatal period due to suicide. And there's also a high risk of a very small number of postpartum depressed moms that develop psychosis. So it's a disorder that just hasn't really been looked upon quite seriously, apart from the fact that, it's something that's known that to happen to some women. So when I looked at research about it a bit more, I realized that it wasn't even recognized as an official disorder until about 1994 in the Diagnostic and Statistical Manual, which is the DSM-5. And this is the manual that's been looked at by, to get your diagnosis from psychiatrists and whatnot. So it's one of those things where it makes me wonder why PPD hasn't really been looked upon seriously. I also know from the research that about 3 in 10 women are not even screened. So in other words, they may be pregnant, they give birth, and that's it. So they don't really get any chance to even be screened for postpartum depression. And we also know that there's a high economic cost, costing in terms of productivity and loss of hours and loss of ability to go back to work, amounting to about US 158 million. There is one medication currently approved for postpartum depression. I'm sure if you're in the field, you would know of the medication from Sage Therapeutics, which is fantastic. But we do see as well that there's a remission rate is probably about 30 to 50%, which is roughly the same as what you would see in classical antidepressants. So this is really a field that is critical unmet need, and it's a field that we should really be focused on. So as I mentioned previously as well, that the gold standard for assessing and screening for postpartum depression is known as the Edinburgh Postnatal Depression Scale, or the EPDS. The EPDS is a universal tool. It's recognized and used pretty much everywhere. And it's a 10 questionnaire and it's scored over 30. And it encompasses questions that will look at not just your depression, but also your suicidality thoughts, which is in question 10, for example. It asks if whether you have any thoughts of harming yourself or your baby. So in order to be deemed as high risk, you need to have an EPDS score of 13 or higher. Or you would have to answer yes to basically question 10 about harming yourself or the baby. Now, the interesting thing about this, or the problem I see here, is that in the DSM-5, postpartum depression is diagnosed 4 weeks after giving birth to a baby. And yet, when we look at the EPDS, the routine screening is usually done at 6 weeks postpartum. So there's really this gap where we aren't really able to diagnose women, because if they were screened and having as a high risk at 6 weeks, at that time it's usually too late. So most of the time in that case, it's most of the women just either not report their symptoms, or they basically just go on with life, not wanting to be, be treated for anything. So that's why we see a really high untreated and undiagnosed rate. And what we see in the literature is that even though we see roughly about 20% of women that are affected with postpartum depression, I wouldn't be surprised if that number is actually a lot higher. So really, there's a need to understand, again, the biology of postpartum depression, just like any other disease. The issue with mental disorders is that because it's so heterogeneous and because of the complexity of the pathogenesis of the disorder, it's really critical that we understand the biology and understand how we can use precision medicine based upon this biology. So one of the things that I'm really interested in is looking at immune targets for precision medicine. And the reason for this is that it's not something that's quite new. Like in the cancer oncology space, we already see that immune checkpoint blockade treatments is very very commonplace. So we know that looking at immune cells is something that has been looked at for a long time. And the reason why there seems to be a link between immune targets and depressive disorders, or at least mental disorders, is that there's a lot of the literature now points towards the contribution of that pro-activated or that hyperactivation of the inflammatory system, which I mentioned previously, that when the stress system is activated in the body, and for certain people, that sensitivity to the stress stimuli doesn't actually go back down when the stress stimuli is removed. And for this reason, there seems to be a connection between your inability to sustain, or your ability to not be stressed in the stress signal, and that activates your inflammation. There's also a lot of evidence showing that when your inflammatory system is activated in a stress state, many other cell systems also are affected, such as your metabolism, things like your glucose metabolism, as well as the oxidative stress that we see in your cells. And the thing with this is that when you basically have immune cells that are affected, they can release cytokines into the plasma, and these cytokines can actually activate different immune cells in the brain, leading to different dysfunctional neuron activation. So really, because of that understanding that we see in the literature, one of the ideas I had for Ketim Therapeutics was that, can we identify or can we predict postpartum depression before it occurs using blood-based biomarkers, based upon our knowledge and understanding of inflammation and how it contributes to major depressive disorder? And this is a really interesting point of view, because we know that, as I mentioned previously, that when women are screened at 6 weeks postpartum, a lot of times it's too late, and we just have too many people that are undertreated and underdiagnosed. So this is something that I was really interested in, is that, and this is not just for postpartum depression. I can imagine in a world where most mental disorders, the onset of age is about the average onset of mental disorders is the age of 32. And we also know in the literature that the highest risk group are people aged between 15 and 29. So a lot of the times we wait for the symptoms to show up. And usually in that time, it's very difficult to be able to identify the cause of the depression or the mental disorders and what's actually correlating to that. We know a lot of early life stress can contribute to that as well. But in the sense of pregnancy, if we are able to predict this before the child is born, for example, we can be able to intervene and do early intervention for the moms, allowing them to seek psychological help if they need, or things like being able to do just psychotherapy with cognitive behavioral therapy. That way, when the baby is born, they have an ability to already be on the track of getting some help before it's too late. So I decided to basically launch Ketim Therapeutics to go along this trajectory of identifying blood-based biomarkers to predict postpartum depression, and whether we could personalize and develop a kit to understand personalized precision medicine and treatment strategies based on a unique patient's biology. In this way, not only are we able to predict risk, we can understand the cause, and we can also do targeted interventions. And blood, relatively, it's relatively non-invasive. We already know that we do a lot of routine blood collections during pregnancy. You get very used to being stabbed by a needle when you're pregnant, so it's not really that difficult to include another vial or another tube of blood to be able to pass on to the pathology lab to be screened for biomarkers that might indicate their high risk of developing postpartum depression. Now, the goal here isn't to replace the EPDS or to replace the work that we need from the psychologists and the psychiatrists, but could we actually make that diagnosis better? Could we actually come up with a more objective way of looking at diagnosis rather than looking at subjective questionnaires, which really can change from day to day? Even for myself, how I feel today is very different to how I might feel next week. So when you use subjective questionnaires like that, it's really difficult to pinpoint exactly how a patient feels. And later on, I'll go through a little bit about my data and show you a little bit of interesting stuff that came out from people's EPDS scores. So firstly, I thought maybe I'll just quickly run through a little bit about biomarkers. So, we know that biomarkers is a measure or indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. We know that biomarkers have actually been looked at in depression, and that they fall into quite a few categories, whether it's diagnostic biomarkers, where you either diagnose a disorder, monitoring, where you can monitor response to medication, say in a clinical trial, response, prognostic, as well as susceptibility or risk biomarkers. And at the molecular level, we know that we can look at many different levels. We can look at the genomics, the epigenome, transcriptome, proteome, as well as the metabolome. And in my situation, or at least in the strategy that we're using for Ketim Therapeutics, we're looking at the proteins. And we're really interested in the proteins because we know a lot of work has been done in the genetics and the epigenetics as well as the transcriptomics. But only because of the advancement in proteomic technology today, as you'll see later on with Olink, we were able to really use proteomics now as a tool to understand whether we can identify, signatures that we weren't able to do before. And we also know that once we identify these biomarkers and how we validate them is really critical, because it's not just about identifying the biomarkers, but then it's about validating this to ensure that if we use these biomarkers to diagnose or to predict, we need to have the qualification from FDA, for example, to ensure that we have the specificity and the analytical precision. And this is something that we hope to do, or we hope to continue on from our work after doing our discovery study with Olink Proteomics. So as I mentioned previously, there are many different methods that has been done in the literature that looks at different technologies. As I mentioned, we can look at genomics, epigenetics, and transcriptomics. And there are other proteomic methods as well as metabolomic methods. And a lot of these methods have been done, and I myself have looked at them. And one of the things that has come up time and time again is a lot of these methods, although they may be specific, they're really low throughput. So we don't have a way of assessing high specificity and high accuracy using high throughput methods until, of course, Olink Proteomics came about. So I was really excited when I actually saw the technology with Olink, because I knew already from my background that there was a lot in proteomics that if the technology caught up with the high throughput and with the accuracy and the specificity that we need, that it would change the field and it would change the way we look at proteins. And this is something that I was really excited about. For me, I have a lot of experience doing things like ELISAs and Western blots. And I remember the long hours, long days, just being able to look at 2 or 3 proteins and feeling really happy when I just have the results from 2 or 3 proteins. So when I used Olink technology and realized I'll be looking at over 5,000 proteins, it was quite mind-blowing for me. And so it was really exciting that I was able to use this for my pilot study. So I guess this is where the exciting bit comes in. This is where I describe a little bit of the pilot study that Ketim Therapeutics has done. So one of the things that I did initially was to partner and to collaborate with the Mother's Hospital, which is Queensland's largest birth hospital. And if you're interested in looking at the study protocol of this study, the reference is there. So basically what this study is a longitudinal study where they recruited Queensland or anyone residing in Queensland who were due to give birth at the Mother Hospital. So the inclusion criteria was basically anyone who was above, anyone who gave consent, and anyone who was going to give birth. So it was pretty broad. So this included, even women who were even included women who basically came from rural communities, which was really important for us. And so we were able to identify, this cohort that had a huge, variation in their demographics and as well as their backgrounds. And this was really good for us. So what we did then is that in this study, they collected screening questionnaires as well as blood samples alongside other biological samples at 24 weeks of gestation, 28 weeks, 36 weeks, and then 6 weeks postpartum. You can see here that questionnaires were given out at 12 weeks, which was the enrollment, where we looked at their demographics, their medical history, their medications, their family history, sleep, exercise, nutrition, and their obstetric history as well. At 24 weeks, when they came in to do their blood tests and their blood collection, they were also given the mental health questionnaires. And these were standard mental health questionnaires like the depression, anxiety, and stress scale, amongst others as well. And then at 28 weeks, they had their bloods collected as well, 36 weeks. And then they were given the same mental health questionnaires again at 6 weeks postpartum. And you can see that the clinical data we were able to get from these questionnaires revolved around their alcohol or drug consumption, their medications that they may have taken or changed since their enrollment, as well as their blood pressure, their heart rate, their weight, BMI, and any pregnancy complications that were discovered at any of those time points. So as part of this Queensland family cohort study that was done, we were able to collaborate with them and be able to identify and use some of their blood samples for our pilot study. So how did we actually end up doing our discovery study? So in the whole Queensland family cohort, we had about 439 women, or I should say families, because the mom, the partner, and and the babies were enrolled between 2019 and 2021. There were some in there that were included that went through the pandemic. So, for the purpose of this study, I actually excluded all the moms that went through the pandemic and had COVID, had been diagnosed or had been screened for COVID. First of all, I looked at the EPDS scores at 6 weeks and identified there were about 311 women out of 439 that completed the EPDS scores at 6 weeks. And so that's about 70.8% hit rate, which is pretty good. We found that there was a high risk of women with PPD. There were about 36 women with high risk, so meaning having an EPDS score of 13 or higher, and a low risk number of women for about 275. So roughly, this was a 30% high risk, which is roughly around the same as what we see in prevalence in developed countries. Of course, I have to say that we don't know if these women are actually diagnosed with postpartum, because I mentioned to you previously that they would have to be diagnosed within 4 weeks. So, we cannot make the assumption that these women would have been diagnosed, but they do have a high risk of developing postpartum depression. So the bloods were taken at 24 weeks, and we saw that we had about 174 women out of these 311 women that had bloods available at 24 weeks. And again, there were about 24 women that had a high risk and 150 women with a low risk. Then we applied our exclusion criteria, which we essentially, like I mentioned previously to you, we took out any women that had alcohol or drugs consumption. We excluded women that had a previous history of mental disorders, family history, had medications that would imply or that they had a mental disorder, as well as any other medications that would confound the results. Essentially, we took out everything. And we found 11 women that basically had what we call the clean cohort. So there were 3 women in there with postpartum high risk of PPD, 10 with a low risk. But what we did is we went back to the data and we also included some women that did not have any EPDS scores at all. So we thought, okay, maybe we can get some of these women without any EPDS scores and we can spike them and see how good our signature is. And so we call it the spike cohort. And then we have some real life samples, meaning we have some women who did not have any exclusion criteria. These are just women that would come through the hospital without any filters added to their biological to their clinical data. And so we had 24 of them, and we labeled them the messy cohort. And essentially, we had about 40 samples that we sent for Olink proteomics. So that's me being really nervous about collecting the samples because I did not want to drop any of the samples. And this is a wonderful picture that our Olink service provider sent to us about the samples going into the machine. So that was a really big and exciting day for me. So this is the results that we got. So as I mentioned to you, we had 24-week blood samples. We looked at their questionnaires and we found 10 participants. And the nice thing about this volcano plot is a t-test that was done between the controls and the postpartum depression or the high-risk women. And when we looked at 5,352 proteins, 3 proteins, protein A, B, and C, came out to be significant. So significant between the moms that had PPD or high risk and the controls. This was really exciting to us because obviously we didn't know if the 10 was enough to give us any results at all. So we weren't really expecting this. And we weren't really even expecting 3. We thought having 1 would be good, but we were very surprised when we saw 3. And one of the nice things that we eventually realized as well is that not only did we get 3 significant proteins, but we got really, really high significance as well. So as you can see from the box plots here, protein A, B, and C, we essentially had a really difference between the first protein, which shows to be highly expressed as compared to the control. And then we had another 2 proteins that were very underexpressed as compared to the controls. And you can see from the table on the right, the next protein onwards, which weren't significant, were quite far in terms of their p-value, their adjusted p-value. And the top 3 proteins were very significant in terms of their p-value. So this gave us a bit of confidence, and we're quite happy to see this. So then when we looked at the heat map, we can see over here that when the controls were put against the mothers with the high risk, we saw a very clean, nice signature come up. And this was really exciting to us. And you can see from the principal component analysis plot here, how correlated and how clustered the controls were to the cases. When we looked at the Olink stat analyzer software and pathway analysis using Reactome and things like that, we were able to show that protein A, B, and C actually converge at a protein known as protein X. So I'm sorry for all the A, B, Cs, and X, but we actually have all of this put in a patent. So we've put a patent application for this, and so we have to keep it secret. So what we found is that the protein X is a novel drug target that we have seen is not currently being used for any psychiatric illness. So it's really exciting to us. And we also realized that A, B, and C could potentially be used as a biomarker panel, not just to look at the risk, but also as a predictive panel towards the response of any drug targets to protein X. And if you look at our AUCs, when we combine the protein A, B, and C, we had an AUC or a sensitivity and specificity of about 0.66. But when we looked at B and C alone, which I mentioned to you previously, was underexpressed in the postpartum depressed cases, we saw an AUC of 0.942. This was really exciting to us because if you look at the EPDS, the gold standard, and depending on the cutoff value of what you use on the EPDS, if you see 13 here, which is what we used, you can see that the sensitivity is about 0.66 and that the specificity is about 0.95. So we're really almost, either having an AUC of at or better than what we see with the golden standard of the EPDS. So this was really, really very fascinating to us. And so we also could look at identifying druggable pockets for protein X, because not only are we interested in the biomarker panels, but we're also interested in novel therapeutics. So one of the tools that we could use is to identify druggable pockets, to identify novel ligand binding sites. And for protein X, we found that one of the binding sites actually had a probability score of 0.9. So we know that that site would probably be a very good site for ligand binding, and that if we do high throughput compound screening, we should be able to find more drug targets to this compound, to this protein. As I mentioned to you, we have 2 provisional patents that just went in based upon our results, and so we're really happy about that. And when we looked at protein A, B, C, and X, we know that protein A, essentially, when we looked at patent databases as well as our FTO, or freedom to operate search, we found that it was 100% novel therapeutic for psychiatric conditions. One drug has already been approved for protein A, but for another indication. So if we wanted to repurpose this drug and try this for postpartum depression, that's something we're looking into. And we've found 4 freedom to operate results for diagnostic methods for neuropsychiatric and neurological disease. So this means we're roughly on the right path. But protein B, C, and X, what's really interesting is that they're 100% novel and not used for any indication, nor have there been any diagnostic biomarkers apart from protein B, which has been used for cancer. So this pointed us towards that specificity and that accuracy that I was talking to you about, that if we were able to find these proteins in plasma, this was really fascinating to us when we're looking at a disorder that we know points towards psychiatric and neurological disease. So if you can see over here as well, that when we look at the heat map of the top 10 proteins, not just the 3 significant ones, a really nice signature comes up. But however, due to our sample size of 10, obviously we weren't able to see the significance of these other 7, these other proteins. But we're hoping that by increasing the sample size and by looking at independent cohorts, this signature will come up more between the postpartum depression high risk as well as the low risk. And just remember, this was done at 24 weeks. So this is really exciting if we were able to find something early in pregnancy that could predict the risk before the baby is born and before postpartum depression is normally screened for. And one of the things that we did was to also look at the spiked cohort, as I mentioned to you previously, and the messy cohort. So we included them into our heat map. And as you can see here, the 3 purple lines that you can see here, these are our clean postpartum depression cohort. So they roughly have a pattern of having that rate or that, that expression with the protein A, and then the lower expression, which is protein B and C. You can see over here, this is an interesting one because we identified a control, so someone who had an EPDS score of 2. But you can see quite clearly from their biological signature that they actually look like one of the cases. So when we went back to look at this participant, we didn't quite know what was happening. But then I looked at her occupation and realized that she's a GP. And I'm not sure if you know, but there's a real thing where GPs aren't really allowed to say that they're having depression symptoms or have a mental disorder because they might get their license essentially revoked. So we see a lot of this happening in the health and professionals who don't report their symptoms for fear of repercussions. So I don't know if that's the case, but this was really interesting to us. If you see up here in the green, this is our unknowns. So these are women that we didn't know what their EPDS scores were. But if you can see from this green box, I'll be inclined to look at this participant and see if this participant is okay based on their biology. But we'll never know because this participant didn't do an EPDS, which again makes sense to me because if you were depressed and had a really difficult time, with your postpartum depression, it's unlikely you'll be well enough to even go to a doctor or do a test or do a screening questionnaire. I know that was the case for me. So that would probably be me. I wouldn't be able to even identify what I was feeling because I was too depressed. And this is why early predictive biomarkers would be really essential as well, so that we don't miss the people that are not screened for whatever reason. Lastly, I just wanted to point towards this blue box here. So you can see this blue box here that is a switch of what we see in the postpartum depression signature, where we have a switch between their colors. And when I looked back at this particular control, which is part of the messy cohort, I realized that this particular participant was on antidepressants. So this was, again, really interesting to us because we know that if this participant was on antidepressants and is acting like a control, then potentially we could even look at participants who are antidepressants and who may need, and we could be able to look at their signature to look at antidepressant response. So this is really exciting to us as well. Again, all of this would have to be validated and verified, of course, but that is one of the, a few of the thought processes we have. So where to from here? So we need to verify our results, obviously. I have identified 2 other birth cohorts in Australia. We're also looking at MDD cohorts now, both locally as well as internationally, whether we can verify a signature in major depressive cohorts, autism cohorts as well, which we know there's a high risk of moms with PPD with children of developing neurodevelopmental disorders later on. We need to validate this study, and we're hoping to do some drug screening, as well as integrate AI machine learning capabilities to enhance our biomarker predictions. And lastly, one of the big goals I really hope to do is to be able to develop a dried blood spot kit to assess mental health biomarkers. And this is really important to me on a personal level because I really care about primary care services in rural communities and accessibility and care for people who just will not be able to have the option to go to a hospital or have the money to basically essentially see psychological and psychiatric services. So this is something that I'm working on in the background as well that I'm really excited to hopefully develop. So I just want to give a huge thank you to all of you for giving me your attention. I also want to acknowledge the Mother Hospital, Olink Proteomics, the Queensland Family Cohort, as well as the Australian Genome Research Facility, which is an Olink service provider in Australia. We also want to thank our participants, collaborators, partners, advisors, and our legal team. So specifically Vicky Clifton, who is part of the Mother Hospital and the Queensland Family Cohort, who's been instrumental in all of this. Danielle and Dr. Elka Kotari, which is our senior OBGYN registrar, who has given us a lot of advice, as well as Desley, Benjamin, and Mark from AGRF, Brenton from Olink, as well as Wayne, Charles, and Ellen, who are my legal team helping me with all my IP protection. I also want to thank my chief scientific officer, who helped me with a lot of the analysis and who's helping us with the AI and machine learning, and our chair and board director, who has a huge experience and background in diagnostics and is going to be helping us as well to develop the governance and the structure that we need to push this company forward. So yeah, I thank you for your time, and I'm really excited that you got to be here.
Thank you. Thanks, Clarissa. This is an excellent presentation and very insightful. It really highlights the importance of this area of research, and I'm really excited to see where your next steps take you for sure. And I would just like to say hi to everyone. My name is Shana Lammers. I'm a field application scientist at Olink. And I'm just going to take a few minutes here to highlight our Olink Explore HT panel and how it can be impactful to you and your research, just like it was here in this example for Clarissa. And so if we think about a brief history of Olink, we really highlight our continuous drive for innovation and desire for biomarker discovery. In 2020, we released our first Olink Explore product using an NGS readout, and it was the launch of Explore 1536, which was a 1500 panel protein. And just within 3 years, we've more than tripled our assay library to expand to just over 5,400 proteins in July of 2023 with the Olink Explore HT panel. But as you can see, Olink's comprehensive offering spans the entire biomarker development spectrum. And so from high multiplex, high throughput solutions, for biomarker discovery, like you saw here today, to customize lower multiplex panels with absolute quantification for biomarker validation and beyond. Oops, where are we here? Sorry, jumped ahead a bit. So in addition, Olink's technology, requires minimal sample volume. It enables you to conserve or save more of your precious participant samples. With Olink Explore HT, just 2 microliters of sample are consumed to measure over 5,400 proteins. Data for this panel are available using NPX values, which stands for normalized protein expression and is a relative protein quantification unit. Our approach allows the detection of thousands of proteins with high specificity while covering a broad dynamic range. And using high throughput workflow and the availability of user-friendly data analysis tools, it enables it really enables you to run projects of any size. And one example is the recent announcement of the UK Biobank, which is launching the largest proteomics study to date, where 600,000 samples will actually be measured using Olink Explorer HT. In addition, forward here. There we go. So this is this really is enabled by a proximity extension assay, or PEA technology, to ensure high quality data with industry leading specificity. The dual antibody recognition of PEA requires antibody pairs to bind to their target protein when in close proximity. Their oligonucleotide tags will hybridize, bind, and amplify, thus generating a signal. If mispaired antibodies bind to the same protein, no signal will be generated, and it will result in negligible cross-reactivity. And the quality and specificity of our technology has been recognized and highlighted within the research community, with more than 2,300 publications in peer-reviewed journals spanning a variety of research areas and study designs. I'm just not sure if it's moving forward. There we go. Sorry, just hung up there for a second. So most importantly, though, is how we've developed the Olink Explorer HT. Sorry, am I having control here, or?
No, I don't think it went to you, so I'm helping you.
Oh, okay, thanks. I was wondering what was happening there. Yeah, I'm trying to take control. There we go. Maybe I got it now. Okay, so here we go. Yes, sorry about that. Okay, so we'll start back here again. So most importantly, it's how we've developed the Olink Explorer HT panel is to really provide deeper biological insights and discovery for your research. And so at Olink, our development strategy involves a rigorous process to not only ensure data quality, but also to ensure assays selected provide biological relevance and for a broad range of research areas. And so during development, we've conducted this extensive validation process to ensure high specificity of the assays selected. In Olink Explorer HT, we actually observed more than 99% specificity. And so this is achieved by that PEA technology I mentioned, where we evaluate each antibody for specificity and ensuring that there's no cross-reactivity with high homology protein pools. Also, we have seen that our data has been incorporated and assessed in large-scale proteogenomic studies, where the genetic data further corroborates our protein assay specificity. And during the design behind Explore HT's library and determining which targets we decide to include, we're really leveraging our growing knowledge in human proteomics and ensuring the design supports new biological discoveries. And so as you can see here from these pie graphs, the protein library in the Olink Explorer HT panel overlaps very similarly with the human proteome. In fact, this panel provides coverage or 100% coverage of the top biological pathways and representation for all major disease areas. Additionally, we're ensuring that the selection of intracellular proteins is present because it's important, as a high percentage of disease-associated intracellular proteins are rarely detected in healthy plasma. But it's still important to include these analyses for novel insights in disease. During the design and the validation, each assay undergoes a 3-step 15-factor analytical verification process to ensure quality and analytical performance. We measure not only the specificity, but many other quality parameters, including sensitivity, dynamic range, and interference, to name a few. Additionally, our validation is freely available, and you can also contact our support team at any point if you'd like to learn more. So not only does Olink biomarker discovery solutions include assay panels like Olink Explore HT, but we're truly here to support you beginning to end with your research and have developed a variety of tools and resources available to support you during the pre-study planning and design, as well as data analysis. And so this tool is known as Olink Insight. It's a very comprehensive suite of user-friendly tools and data sets that help you to further expand your knowledge in proteomics. It's free of charge, and you can and it's there to assist you at every step of the way. So whether it's from project planning, design, to post-study, post-run data analysis, as well as offering the ability to create reliable and publication quality insights. We've seen Olink Insight be used in many labs of all sizes to uncover new proteomic findings. So it's really exciting to see where this will take us. And just to highlight a few of its features, I'll just touch on a couple of them here briefly while we have a few minutes. But the Pathway Browser tool can aid in understanding connections between protein and human biology. By creating curated protein lists, we can look through the Pathway Browser to see what biological pathways they're involved in and other proteins that may be associated. We also offer tools that help you with your research question, or to put your research question into action. And so here we see the Disease Atlas, which is an open access collection of proteomic profiles for critical diseases. We have a study size calculator that can help enable you to better understand the power of your study based on your participant size, sample size, or groups, as well as the proteomics disease risk data story, which I'll touch on here a little bit more in a minute. Once you've completed your testing and have Olink data in hand, we have resources and tools to evaluate, analyze, and make sense of your data. Our Stats Analyzer tool is a user-friendly web application that includes commonly used statistics to help you begin your journey on the data analysis. We also have data on normal ranges for proteins, which explores individual variability of approximately 3,000 protein assays from 3 cohorts and across 3 continents. And so this is also broken down by key characteristics such as sex or age. And then lastly, the Publication Explorer. So here it's a great forum to search abstracts for your proteins of interest using text mining of more than 30 million publications. And just briefly, I just kind of want to highlight an example here of how one of these tools can really be used and how it showcases the possibility of Olink Explore data in this example with UK Biobank. So looking at proteomics of disease risk, we're combining protein measurements with longitudinal healthcare data from over 50,000 individuals. And from here, we're able to estimate the effects of nearly 3,000 proteins on the future of more than 100 diseases. Within the story of Olink Insight, we provide summaries and overviews, as well as detailed presentations highlighting some really interesting medical results. The data set is also available for download as well for your review. And so you can see how a resource like this can really be an inspiration on how proteomics data can be used at any scale. And so the goal here is, with these online tools in conjunction with Olink's comprehensive protein library, is to really create an ecosystem of products and services that help drive biomarker discovery within the research community. We want to provide a seamless Olink experience that supports you and your proteomic research end to end. So I'll just close off quickly here and open it up to discussion and questions with the group. Thank you for your time.
All right, thank you very much for your presentations. We do have a couple poll questions that we want to take care of before we get into the Q&A. While we're doing that, feel free to send in any more that you have. The first poll question we have is, do you think biomarkers play a crucial role in understanding mental health and managing treatment strategies? Yes, unsure, or no. And we'll give everybody just a few moments to answer that. And as those answers are coming in, any of the questions that we don't get to today, we will follow up via the email you used to register. I'll just give another minute or two or another few moments on this one. All right, the next question we have here is, what aspects of biomarker research and mental health research interest you the most? Early detection of conditions and monitoring treatment responses, exploring new therapeutic targets, or understanding biological mechanisms? Give that one a few moments. All right, and the last poll question we have before we'll jump into the Q&A is, how familiar are you with affinity-based proteomic profiling technologies like Olink Explore HT? Very familiar, somewhat familiar, not familiar. And while that last one is up there, I will go ahead and start with our Q&A. Let's see here. First one we will do is, was PROTX measured as well during Olink or later by some other means? And oh, it looks like Clarissa had a disconnection, so we'll give her a moment to join us back. There she is. Are you able to hear us, Clarissa?
Can you see me now?
We can hear you, but we
I can
can't
hear
see
you.
you.
Can you hear me?
Yes, we can. Can you hear us?
I can hear you,
Okay.
but I'm not sure you can see me. I don't have the button that allows me to click.
Yeah, we're not able to see you, but we can hear you. There we go. We'll try that, and you can hear me now.
Yes, technology.
Okay.
It's good when it works, but
So,
when it doesn't.
so we'll do just a couple questions here to wrap up, and any questions that we're, like I said, we don't get to, they will follow up via the registration email that you provided. So this first
Yes.
one here, yes.
Yes, I'm happy for anyone to put questions to me, even through my email address at Ketim Therapeutics. More than happy to take the conversation outside as well.
Okay. So this first one here was PROTX measured as well during Olink or later by some other means?
So that's an interesting question. So I did go back to the Olink's list of proteins. We did see protein X there. However, the protein X was not listed as one of the significant biomarkers. However, we're not looking at it as a biomarker. We're looking at it as a novel therapeutic. So the thing that's nice about our data is that A, B, and C came up as highly significant. And through bioinformatical, pathway analysis using software such as Pathway Comments and Reactome and string databases, we were able to identify that the three proteins were actually connected to protein X. So protein A was connected to protein X by 2 degrees, meaning it was regulated by another protein that is then regulated by X. But B and C were directly, regulated by protein X and vice versa. So no, we didn't. We saw it on the list, not as a biomarker, but we're looking at it as a novel therapeutic paradigm that we can work on.
All right. Let's see here. In addition to the 3-7 patients' controls, have these findings been confirmed in other patients/controls?
So that's the nice thing about our data. So when we included some of the messy and the spiked cohort, we did find this signature state for some of them, not all of them. So therefore, we know that with some of the confounders that came from the messy cohort, in other words, we didn't exclude anyone that had, say, for example, pregnancy complications. We didn't exclude anyone with medications, etc. And we found that for some of them, the signature stayed the same. So in other words, we wanted to test the robustness and the specificity of the biomarkers. And we found that was true for some, but not all. So, we know that the heterogeneous nature of mental disorders is what makes it difficult. However, that's why we're thinking with tools like, artificial intelligence and machine learning, we would be able to integrate all these other variables that would then give us a higher accuracy and a higher enhanced prediction score. So we do need to verify this in an independent cohort, which is what we're doing right now. So we've identified 2 more other pregnancy cohorts that we would like to validate this signature. And then we're hoping to be able to then validate this in other depressive cohorts as well.
Thank you. So we've got time for one more. So we'll go with, were there any findings from the study that surprised you or challenged existing paradigms about PPD?
Yeah, so I think PPD as a whole in general, together with other affective psychiatric disorders such as major depressive disorder, the difficulty about these mental disorders is that for the longest time and even up till today, we still use questionnaires as our gold standard. And so what really surprised me was that we were able to find biological markers in the blood that helped us to identify and these participants that had screened positive in their questionnaire. At the moment, there is no such blood tests available clinically. So there is a lot of research showing that you can use blood-based biomarkers using other methods, but none has been clinically validated to date to be used as a clinical utility tool. And that's where Ketim Therapeutics comes along, is that we are identifying these biomarkers. It is it makes sense when you look at the literature. It is coherent with what we understand about the pathogenesis and the neurobiology of depression, and the signaling pathways that we identified makes sense to me in the terms that is what I know from the literature on major depressive disorder. But no one has clinically validated it. And when I say validated, I mean go through regulatory approval. In the US, it would obviously be going through your FDA biomarker qualification. These are things that we have to do externally, and we're on track on doing all of those things.
All right, excellent. I want to thank our speakers again for their time and important research, and we'd like to thank LabRoots and our sponsor, Olink, for underwriting today's educational webcast. I'd also like to thank the audience before we go today and for the questions that we received. Any questions we didn't have time for today, and those that will be submitted during the on-demand will be addressed via the contact information you provided at the time of registration. The webcast can be viewed on demand, and LabRoots will alert you via email when that's ready for replay. We do encourage you to share that email with your colleagues who might have missed today's live event. So until next time, thank you very much and goodbye.